Hello everyone, it is a great honor to present our research to you. Pangenome variation analysis uncovers the impacts of SNP-independent structural variations in modern European cattle breeding. Let me begin with the background. European cattle are central to global beef and dairy production, with key genetic variants offering valuable resources for trait improvement. However, SNP-based studies fail to capture part of the functional variations, resulting in missing heritability. Pangenome analysis enables more comprehensive investigation. Some SVs cannot be tagged by SNPs. Their distribution, origin, and functional roles remain unclear. First of all, we constructed representative European cattle SNP plus SV reference panel via kinship filtering that provides significantly more insertion variance than previous studies. Sequence similarity analysis revealed that 70% of insertions correspond to known transposable elements, enriched in BOVB and L1BT families. We identified SNP and SV hotspots. Genes in these regions are enriched in immune and olfactory functions. Key genes affected include superintendent3H and RUNX2, involved in skeletal development. We classified SVs based on linkage disequilibrium, LD, with nearby SNPs. Tagged SVs were defined as those with R² greater than or equal to 0.6, and non-tagged SVs as those with R² less than 0.2. We found that 19.3% of SVs were not tagged by SNPs. Non-SVs have lower allele frequencies and are enriched at chromosome ends. Tagged SVs are mainly located in intergenic and desert regions, whereas non-tagged SVs are enriched in promoters and active regulatory elements. Functionally, non-tagged SVs are associated with meat and milk-related QTLs. PCA showed that non-tagged SVs effectively distinguish European beef and dairy cattle. Using FST analysis, we identified strongly selected non-tagged SVs enriched in meat and milk-related genes. Between dairy and beef, only 50% of candidate SVs can be tagged by SNPs. But, when comparing dairy versus beef cattle, over 85% can only be detected by non-tagged SVs. For example, a non-tagged SV within the COMMD10 gene, which disrupts local LD structure. This variant is highly frequent in Xuelei, beef cattle, but nearly absent in dairy cattle, and significantly downregulates COMMD10 expression in liver tissue. To explain why non-tagged SVs enrich at chromosome ends, we examined their distribution with recombination and LD blocks. Non-selected non-tagged SVs enrich at ends due to high recombination disrupting SNP linkage. Among selected non-tagged SVs, 44% reside in mobile elements enriched in young MEI families. LD around SVs was notably reduced in European cattle compared to Northeast Asian breeds, suggesting these non-tagged SVs are recent, recurrent mutations shaped by population-specific selection. Although 50% SVs can be tagged by SNPs, SNP genotyping alone is insufficient. Among 6,000 SNP or SV haplotype blocks, 40% gained haplotypes after adding SVs, especially in low LD regions. SVs create breed-specific haplotypes on CHR20, 2 SVs branched from a major haplotype, with one unique to Angus. For example, an insertion on CHR3 in TRAF3IP1 drives breed differentiation and regulates muscle and liver genes. Using a European cattle SNP and SV panel, SV imputation was accurate across breeds. GWAS on 5 traits identified 30 significant signals, corresponding to 26 SVs annotated to 14 genes. An SV near DGAT1, overlapping ADGRB1, contains transcription factor binding sites linked to milk fat percentage. Another SV within a BTLTR1B mobile element, associated with milk yield, is enriched in European dairy but deleted in beef breeds. In this study, we constructed a European cattle SNP and SV imputation